Hello and welcome back. The topic for this video is going to be the genetics of cancer. So we're going to talk a little bit about what goes wrong at a DNA level with cancer. Okay? Cancer is going to be uncontrolled cell growth. So the biological process that we're talking that goes out of whack here is mitosis. So the process of mitosis is no longer being properly controlled. All right? And what we've got with cancer, we are going to have oncogenes, and we are going to have genes that are tumor suppressors. So there's kind of two ways that we can get the genetic regulation of mitosis to go awry within the cell. And it's a little bit simplified for me to just say there are two ways. There are actually more ways than that, but the ones I'm going to focus on in this video are oncogenes and tumor suppressors. Okay? Oncogenes are going to be signals to the cell to tell that cell to grow. Okay? And the key about oncogenes, okay? An oncogene is a mutated version of a proto-oncogene, okay? A proto-oncogene is going to be a gene that normally is involved in cell growth. The difference between a proto-oncogene and an oncogene, we get a mutation that happens. So we get a change at the DNA level, usually, is what's going to happen. And in terms of those proto-oncogenes being mutated to oncogenes, you can either end up having that oncogene making more of the protein than it normally would. So that's going to be the signal that tells cells to grow when they're not supposed to. All right? And I'm going to have tell cell to grow, but it tells cells to grow when the cells aren't supposed to undergo mitosis. All right? That's the key. All right? So you can have the normal protein made, but it's made in higher levels. You can have a mutation that causes essentially the protein to be hyperactive. Hyperactive meaning it functions more than normally and tells cells to grow when it's not supposed to. All right? But when we're talking about oncogenes, oncogenes are going to be the mutated version of a proto-oncogene. And that's key because those proto-oncogenes are what normally tell our cells to grow when you have something like a scrape of the skin. So there are going to be proto-oncogenes that are going to be activated in your cells of your skin to make more skin cells to repair that wound. So that would be a normal process. When you have a mutation from the proto-oncogene to the oncogene, now we've become entering the realm of being abnormal, okay? So you get cell division when you're not supposed to, okay? An example of a proto-oncogene that I've got in the PowerPoints is the protein RAS, okay? That RAS protein is going to be one that is critically important in regulating the signals that tell a cell that it's supposed to undergo cell division. When the RAS protein is mutated, okay, that's going to be one of these signals to tell the cell to undergo cell division, to undergo mitosis. All right? Kind of on the flip side of that, we've got genes that are tumor suppressors. So this is also going to be at the DNA level. Those tumor suppressor genes normally stop or halt cell growth. Okay? So you've got kind of a balance going on in the body. If a cell gets mutated to an oncogene, it can be that as long as there's a normal tumor suppressor, that tumor suppressor says, no, stop, we're not supposed to divide, okay? And then what happens is there's going to be a whole nother pathway that's activated that'll kind of take that cell out of commission. So those tumor suppressors have the ability to tell cells kind of to implode and die, okay? Because they're there to as kind of a checkpoint so that abnormal growth doesn't happen, okay? The problem is if we get a mutation in a tumor suppressor gene, we no longer have that stop sign or the signal for that oncogene to stop dividing, okay? And what that kind of brings us to is with cancer, you've got the multi-hit hypothesis or theory. Which means you have to have mutations in more than one gene to lose the genetic regulation that's associated with the cellular process of mitosis. So that multi-hit hypothesis basically means that when a cell has a mutation, typically just one mutation is not enough to trigger the abnormal cell growth associated with cancer. You typically need to have a proto-oncogene mutated to an oncogene, and you also need to have the loss of a tumor suppressor gene. So when you get the mutation of a tumor suppressor gene, the mutation that occurs with the tumor suppressor gene is the tumor suppressor gene no longer functions. All right? So with cancer, it's not just one mutation typically that gives rise to the process of cancer. You've got multiple mutations that have to happen. That's that multi-hit idea, all right? Our tumor suppressor genes have to be mutated so they no longer function. The example of the tumor suppressor gene that I have in the PowerPoints is P53, okay? So really it's a protein that's involved in halting cell division, okay? So you can kind of think of P53 as the brakes, okay? Our oncogenes like RAS, that would be the accelerator, all right? Step down on the accelerator, but if we've got an active tumor suppressor, that's going to stop things, prevent the cells from growing out of control. But if we've got an accelerator going and we no longer have our brakes, those cells are going to start to divide out of control. Okay? So in terms of the genetic regulation of cancer, it's really interesting how different genes come together to regulate essentially that process we talked about weeks ago, mitosis. Okay? Because you only want to have cells undergoing mitosis when you need to replace other cells or when you have damage. All right? So the other thing that I like to talk about when I talk about cancer, okay, is tumors, okay, or the idea of the tumor, okay. A tumor is, we'll call it a collection of abnormally dividing cells. Tumors can fall into one of two categories, okay? Tumors can be benign, meaning it's not yet cancer, okay? An example of a benign tumor would be a colon polyp. So you get an abnormal growth of cells inside the colon, but they haven't moved from where they started in the colon. The things about benign tumors, you can surgically remove, the benign tumor, and then that set of cells, because they're no longer present, they won't give rise to cancer. Okay? Cancer is kind of the next step from benign in some cases. Some cells can remain in a benign stage. They never reach the cancerous stage. Other cells, it's kind of a stop along the way to becoming cancer. So when we're talking about cells being cancerous, they now have the ability to move to other places. So those cells have metastatic potential, meaning they have lost the ability to kind of stick to the cells around them, and they're now able to leave that primary tumor and go other places in the body. When that tumor progresses from benign to metastatic, the metastatic tumor is the one that we consider cancerous. All right? Hopefully that makes sense. If you have any questions, please let me know.